Hey, good morning, everyone, and welcome to the August 2021 Wild Science webinar. And we've got another great program lined up for you this month. Thanks for joining us on this August 2nd. We're going to have Blake Sassy talking about trends in Arkansas bat populations, and Tate Wentz talking about cyanobacteria blooms in Arkansas. Would like to remind everybody at this point that this is being recorded. Please mute your microphones. At the end of each presentation, you'll have 5 or 10 minutes to ask any questions you have. You can also type questions into the comment section, and we'll get to those at the end of each presentation. First up this month is Tate Wentz. Tate received a Bachelor of Science degree in Fisheries and Wildlife Biology from Arkansas Tech University and a master's degree in biology from Arkansas State. Before joining Game and Fish 2 years ago, Tate spent 11 years with the Arkansas Division of Environmental Quality as an ecologist coordinator over the field and research program in the Office of Water Quality. In that role, he investigated effects of nutrient enrichment on water quality and aquatic life, and for the purpose of developing water quality standards recommendations. In that capacity, he also co-founded the Arkansas Harmful Algae Working Group in 2015. Tate has spent his entire career studying the ecology of Arkansas streams, rivers, and lakes, and looks forward to continuing that with the help of citizen scientists in his role on the stream team. Tate Wentz, the floor is yours.
Thank you, Trey. Just gonna change my background real quick.
I'll fill the space by saying that Tate also makes the best refried beans in Arkansas. I really enjoyed those at Brian Wagner's retirement party, and I was supposed to be getting the recipe, but I guess it got lost in the mail somewhere, Tate.
Oh, sorry about that, Trey. I hadn't got that message, so I'm definitely working on getting that.
Thank you.
Well, I appreciate the introduction, Trey, and I appreciate everybody's interest in the cyanobacteria and attending today. So as Trey mentioned, I spent a good portion of my first part of my career working on nutrient-related issues and dealing with cyanobacteria blooms. But some may be asking, how is Stream Team related to working with cyanobacteria blooms and nutrient-related issues? And it's really more, we're involved from a preventive standpoint. So our mission is to improve fisheries in Arkansas by involving citizen scientists, stream conservation, and watershed protection. So we work with volunteer citizen scientists to train them and educate them on the importance of water quality. And we also work with them to monitor water quality trends through time and space, including nutrients. We work with watershed groups across the state to inform and promote water quality advocacy in local streams and rivers. Lastly, we work with landowners across the state to develop aquatic habitat restoration plans, primarily stream bank stabilization plans, to help reduce sediment and nutrient inputs into our streams and rivers. And so part of this, we also work to enhance the riparian corridor or stream buffers along those stream bank stabilization projects. So again, it's more of a preventive standpoint than actually dealing with the cyanobacteria themselves. So I've mentioned nutrients several times, and so that's really at the root of the cause for cyanobacteria blooms in Arkansas. There's two primary sources: point sources, which are the regulated endpoints, say an end of pipe or an effluent canal, from a wastewater treatment plant or industrial source like a factory. The other type are unregulated non-point sources, which are more difficult to isolate and pinpoint, but we do know they come from neighborhoods, city streets, parking lots, septic tanks, and agricultural fields. This is where most of our nutrient issues arise. While nutrients are important to the aquatic food web, when in excess, they can increase the phytoplankton production, which can increase food sources for zooplankton and forage fish such as shad, and ultimately can increase game fish. But when an overabundance of nutrients occur, cyanobacteria can outcompete the phytoplankton and disrupt this food chain. So I've mentioned several types of algae already, and so phytoplankton is generally a good type of algae. And generally, this can be a managed outcome. Our fisheries management biologists in low reservoir or low nutrient reservoirs will often add fertilizer to stimulate or kickstart the phytoplankton production in hopes to increase the food base for forage fish and hopefully increase the production of game fish. These are generally have a greenish tint and no smell associated with these. Nuisance bloom are dominated by green algae and are generally aesthetically displeasing because they can occupy the entire water column. Because of this, they can have an effect on aquatic life. This is primarily through increased or decreased dissolved oxygen, but as these organisms decompose, they will consume all of the dissolved oxygen. At this point, you'll have some pretty significant odor issues as well. Lastly are the harmful algae. In Arkansas, these are dominated by cyanobacteria. The reason they're harmful is because they can produce and release cyanotoxins. These toxins are harmful to us, our pets, and aquatic life. So what are harmful algal blooms as a whole? Again, I've mentioned cyanobacteria or blue-green algae, but there's two other types, particularly red tides you may have heard about coming out of Florida. At present, there's 100 miles of western shoreline in Florida currently affected by a red tide. At the end of July, 1,700 tons of fish and marine life have washed up on Florida shores due to red tide events. In the Southwest U.S., there's the golden algae. Again, a prolific spawner and producer of toxins can be devastating to aquatic life when they occur. They have occurred in Southwest Arkansas, but not with any significant frequency. They tend to prefer higher salinity waters. So again, we're really focused in on cyanobacteria, and they are naturally occurring in our creeks, rivers, and streams, but it's only when nutrients are in excess of natural background concentrations that cyanobacteria can outproduce the phytoplankton. The exact mechanisms that cause cyanotoxins to be released at any time during the bloom are still being investigated. However, we know that during certain times when the cells are stressed, they can lyse or break open and release their toxins. There's 5 common toxins that we monitor: saxitoxin, anatoxin, lingbiatoxin, microcystin, and cylindrospermopsin. The latter two have the most robust data sets and are also accompanied by regulatory drinking water limits and recommended criteria for protection of recreational swimmers. So you still may be thinking, I've never heard of a HAB, or I don't even know what this is. This is all new. Well, the reason might be because oftentimes they're reported out of local news sources, newspapers, and really only focus in on local swim beaches. So they may never make national news. But as of June of 2021, EPA has documented blooms across all 4 corners of the United States. These occur globally too. Nutrients are a global issue. But two national cases that you may have heard about was Lake Erie, seen here in 2019, and the algal bloom is the green iridescence color. In 2014, the city of Toledo drinking water source out of the lake was compromised by a significant cyanotoxin bloom. At that point, 500,000 people were told the water was non-consumable and to not even touch it because the concentrations of cyanotoxins coming out of the tap were so high. Lake Okeechobee is Florida's largest freshwater reservoir. At any one time, cyanobacteria bloom can cover 70% of the lake's surface area. To complicate matters, the water management strategy is oftentimes water from the lake is discharged into the ocean. So nutrient-rich, cyanotoxin-laden water is discharged into the ocean, which is feeding a red tide. But we'll come back to these 2 examples in a little bit. So as far as cyanobacteria in Arkansas and cyanoHABs, in 2014, Lake Nimrod in West Central Arkansas had a significant bloom. In over a 3-week period, cell concentration reached a maximum of about 100,000 cells per milliliter. What this left state agencies responding to this, kind of scratching their heads saying, this is new, this is different, how do we respond to this? So ultimately that resulted in the formation of the Arkansas Harmful Algal Bloom Work Group in 2015. The sole mission of that group was to improve intra-agency communication and develop response plans for how agencies should be responding and managing HABs into the future. So an output of that was the Harmful Algal Bloom Management Plan. This management plan develops recommendations for resource managers when and how to issue advisories and lake closures. So the recommendation is that if cyanobacteria blooms are present, that an advisory be issued and signage be placed up around the lake. Warnings and lake closures should be issued if microcystins are present at or greater 8 micrograms per liter, and Cylindra spermopsin is present at 15 micrograms per liter or greater. So since the release of the management plan, Arkansas Division of Environmental Quality has been tracking and monitoring HAB events across the state. So indicated by the squares are HABs that had microcystin greater than 8 micrograms per liter. Circles indicate events less than that. In each instance, red indicates more than one event happening at a single lake. So what the data suggests is that areas of higher population densities, such as central Arkansas and northwest Arkansas, have a higher frequency of HAB events, particularly with those greater than 8 micrograms per liter. Also, since the release of the management plan, Arkansas Game and Fish Fisheries Division has had to issue one advisory for Lake Overcup. While a cyanobacteria bloom was present, an advisory was issued, and only one sample from the lake in the early part of the bloom exceeded the microcystin concentrations for issue a warning. However, during the 41 days of the advisory, no samples were above the 8 microgram per liter recommended value for issuing a closure. Arkansas's reservoirs are diverse in their uses, from domestic water supply, recreation, irrigation, and fishing. So the impacts of cyanoHABs on those uses will be equally diverse. So the remainder of the presentation is really only going to focus in on the potential impacts on our fishing industry. So going back to the Lake Erie example, a group of researchers wanted to understand the potential impact of algal densities on fishing license sales through time and space over a 3-year period. In order to do this, they evaluated license sales from every county along Lake Erie's border for that 3-year period and compared that to the annual algae densities. So for that 3-year period, fishing license sales dropped between 5 and 11% when cyanobacteria cell counts were greater than 20,000 cells per milliliter. To put that into context, the Lake Nimrod event was 100,000 cells per milliliter over a 3-week period. So that 5 to 11% drop in license sales equated to about 3,600 fewer licenses sold over that 3-year period. Approximately $2.2 to $5.5 million were lost in fishing-related revenue during that 3-year period. So still on Lake Erie, another group of researchers wanted to know, do anglers even care about harmful algal blooms? And to answer that question, they had 3 objectives. First being to assess whether Lake Erie anglers were willing to pay for clean water. Second being whether there's an economic impact to U.S. anglers. And lastly, to measure the magnitude of impact on the Lake Erie's fishing industry. So not surprisingly, anglers do care about clean water and are willing to pay for it. So much so that they're willing to pay $8 to $10 per trip for 1 less mile having to travel through a HAB to reach their destination. They're also willing to pay between $37 and $51 per trip for 1 less hour needed to catch 1 more walleye. But most impressively, they were willing to pay between $65 and almost $100 per trip for improved water quality clarity. So part of the study also wanted to address anglers' fishing habits. And the first part of that question was, are they even aware of what a HAB is? And 84% of Lake Erie anglers said yes, they were aware of what a HAB is. But how did HABs affect their fishing habits? And 53% said they changed fishing location due to a HAB. 16% said they didn't even go at all. 35% said it had no effect. And 34% reported less time fishing because of a HAB. So the Lake Okeechobee example, over 2018 and 2019, when there were significant cyanoHABs on Lake Okeechobee and red tides in the ocean, a group of researchers from South Florida wanted to ask boat retailers and marina surfaces how this affected their businesses. And during that time period, boat sales declined by 75%. Fishing supplies declined by 56%. Local businesses reported a 28% reduction in annual revenue in 2018 and 15% in 2019. So just to wrap things up, Stream Team is working across the state to improve riparian corridors or stream buffers that will help reduce nutrients. So this is a preventive strategy. We do know that algal blooms differ in size, shape, and impacts across the state and nation, but this is, again, due to nutrient concentrations, and as nutrients increase, we will see an increase in severity, frequency, and duration of harmful algal blooms across the state. So, and also, we do know that as cyano blooms do occur, they can have a significant impact on the economy of our fishing industry in Arkansas. But again, we're working to try and prevent nutrients from even reaching the water and hopefully prevent cyano blooms from occurring and increasing the water quality longevity of our reservoirs. If you'd like to learn more about cyanoHABs, I recommend you go to the epa.gov/cyanoHABs website, where you can find more basic information on cyanoHABs and HABs in general, HAB management, prevention, control, and treatment, the current advisories and closures, the state of the science and predictive modeling, and evaluate state monitoring programs. And with that, I will take any questions, and I appreciate your time. Thanks.
All right. Anybody have follow-up questions? I know I have one, and it's kind of specific to the map you showed of the squares and dots in Arkansas. And just out of curiosity, there was a concentration linear in Northwest Arkansas. What body of water or bodies of water is that? Just curious.
Sure. The red square will actually be Lake Fayetteville. The red circle, I believe, is a small neighborhood reservoir within the city of Fayetteville. And I would have to, the green dots may be Elm Springs and potentially Bella Vista. I would have to reach out to the Division of Environmental Quality to find out for sure where those are.
That answers. I was just trying to make sure it wasn't like the same body of water, different outbreaks over time. So that was where I was coming from. Thank you. Anybody want to follow up with other questions? All right, Tate, thank you so much. Appreciate that presentation. Great job. Next up is going to be Blake Sassy. Blake is our first repeat performer here on the Wild Science webinar. He was part of our very first Wild Science webinar back in January. Blake, of course, has been our non-game mammal program leader since the inception of the program in 2000. Blake got his Bachelor of Science degree in Wildlife Management from the University of Missouri in 1991. And his master's degree in wildlife management from the University of New Hampshire in 1995. Prior to coming to Game and Fish, Blake worked as a wildlife area manager for the Florida Fish and Wildlife Conservation Commission. That was 96 to 99. Now Blake focuses his work on the management of endangered bats, but also works with other non-game mammals and also serves as our fur bearer biologist. And in recent years has concentrated on studying the plains spotted skunk, which is being considered for protection as an endangered species. Blake Sassy will take over now with trends in Arkansas bat populations. Blake,
okay,
take it away.
Appreciate it. So thanks everyone for spending some time this morning to learn a little bit about what's going on with the bats here in Arkansas. We're going to start off with a little refresher on Arkansas bat biology. They are our only flying mammals, and one of the most unique things about them is that they can live for a very long time. One of our species, the little brown bat, has been known to live as long as 34 years. And this capability of living a long time, which 34 years is a little bit of an exception, is one of the things that has helped some of our species recover from previous problems that they've had. And we'll discuss that in a second. But all of our bats here in Arkansas eat insects, and that's pretty common for most bats around the world. But there are about 30% of the species that will eat fruit and nectar. Now, when people think of bats, they don't often think of them living in trees, but most of our bats do live in trees during the summertime, either in a cavity in the tree or a crevice, sometimes underneath loose bark. Some, like the red bat there, will just hang out in a clump of dead leaves on the end of an oak branch. But in the winter, about half of them will hibernate in caves. but only 2 of our bat species will use caves year-round. Now, of the 16 species of bats we have here in the state, 5 do have some form of extra protection provided beyond what we normally have for our other non-game animals. The top 3 were listed as federally endangered back in the 1970s, primarily due to disturbance caused by people that caused them harm while they were staying in caves during the summer or the winter.
Hey, Blake.
Yes,
Blake, we are not seeing your presentation.
we're not? Oh.
We are not.
Let's try that again.
There we go.
Okay.
Bingo. Thank you,
okay.
Blake. Sorry.
So yeah, those top 3 are federally endangered bats I mentioned. The bottom 2 are more newly protected. The northern long-eared bat, which we'll talk about in a second, was federally threatened and is also state endangered here in Arkansas. And the little brown bat is state endangered here in the state. The biggest threat that's facing bats right now is white nose syndrome. And this is caused by a fungus, which is totally unknown to science prior to 2000. And you can see it on that bat there around its nose and on its ears and on its forearms. It causes this, white growth that is easily, seen in its more advanced stages. So this fungus was first seen by people up in New York in 2006, although there is some evidence it had been present in the United States for 5 or 10 years before that. But the first time bat biologists noticed it was in a cave here in New York. Since then, the fungus has been observed spreading up and down the Appalachians and the Northeast, over into Tennessee, Missouri, and Arkansas, and until fairly recently had been confined to the eastern United States. But a few years ago, there was a big jump over to Washington, and since then it's shown up in more isolated spots in California. It's also kind of spreading up and down kind of the central Great Plains area. The reason or the ways this fungus gets around are primarily, from bats contacting other bats. Bats like to cluster together, and that's just a perfect way to transfer, fungus or their spores from one individual to another. But we do suspect that people can spread this fungus around as well. That's probably how the fungus got to the United States from Europe in the first place, which is where it appears the fungus originated from. People go in caves, they can get the fungus or its spores on themselves or on their gear. And if they go to another cave, those spores could fall off and start to grow there. And, we think that's how it made those big initial jumps. But once it got to the United States, probably most of the transmission just came from, going from bat to bat. But the human transmission route is difficult to deal with because it's incredibly difficult to decontaminate your gear from going in a cave. It can be done, and folks like myself that work in caves do it all the time, but getting the general public to decontaminate can be tricky. So it causes this white growth on the bat. So why is that a problem? Well, the most current theory is that this fungus kind of irritates the bats, and it's getting on them in the caves in the winter while they're hibernating. They're basically trying to sleep through that winter, and the fungus causes them to wake up more often than they normally would throughout the winter. And every additional time they wake up, it causes them to use more energy to do that. And if they wake up enough times, they use up all their energy, and basically they starve to death. And you end up seeing big piles of dead bats on the floor of some caves. And for some species, this has caused like 90% or more mortality at some particular sites. And just because of white nose syndrome, the northern long-eared bat was listed as federally threatened in April 2015. It seemed like an almost 99% decline in the Northeast. This was a species that was incredibly common across all the eastern United States. They were all over the place. You'd catch a lot of them. And just because of white nose syndrome, their populations just dropped off a cliff, and they're being listed because of that. And there are several other species that have been impacted that are also being considered for listing as well. And theoretically, pretty much all cave species are potentially vulnerable, but in practice, we're not seeing that same sort of impact on all of them. But when we're trying to look at cave bat populations, we'll go in the caves, usually in the winter when the bats are gathered and hibernating, and we'll basically count them. If you've got just a single bat, such as this big brown up here in this crevice, obviously it's pretty easy to count them. But a lot of the bats we're concerned with do form clusters, like these Indiana bats in the top right. And Indiana bats cluster very densely, up to 400 bats per square foot. So when we're trying to estimate their populations, if it's close enough, we might get out a measuring tape and just kind of measure the size of these clusters, then multiply it by 400 per square foot to get our estimate. So that might be 800 bats in that photo, roughly. That works for smaller clusters. Now, if you go in a cave that has gray bats, such as this cave here, some of those caves can have hundreds of thousands of bats. We have several in Arkansas that are well over, 100, 200, 300,000 some years. So there's bats everywhere. And that takes a little bit more effort to try to estimate the size of these clusters. And again, we kind of use a standard multiple to come up with a population estimate. Now, there are some bats that we'll try to monitor in the summer. And one of the ways we do that is watch them fly out of the caves. And we'll just kind of count the bats as they come out. Usually we'll use some night vision goggles to extend the period in which we can count them. And there are some fancier methods you can use now of recording them with infrared, and there's some automatic counting systems you can use to try to get a more precise estimate. But that's the basic way we're gaining this information. So with the surveys we've been doing, we've been watching this white nose syndrome spread throughout Arkansas. It's first found here in 2012, and since then we found it in almost 90 caves. You'll notice that the chart stops in 2019, and that's because in 2020, in that winter, we weren't able to do any bat surveys. With the coronavirus coming to the United States, we're very concerned about possibly transmitting that virus to our bats, which don't have it naturally. And so just to play it very safe, most state agencies decided not to do their winter bat surveys in 2020. So we don't have really current up-to-date information on a lot of our bats. But as you'll see, the white nose has been spreading pretty rapidly and up to almost 90 caves now. But keep in mind, that's only at caves where we go and look for bats. There's well over 3,000 caves in Arkansas. So the sites where we've documented it are relatively low. But I think we can safely assume it's much more widespread than what we've seen just looking in caves where there's relatively large numbers of bats. And to put that in context, of the caves that we monitor for these 4 species, roughly 50 to 65% of those caves have white nose syndrome. So there's still a lot of unaffected caves out there, or at least ones where we haven't been able to confirm that the fungus is present. So we're still a little bit on the leading edge of white nose syndrome. Of course, over time, we do expect more and more caves to become impacted. But to start to look at some of the numbers here for what's going on, the tricolored bat, which is a bat that was very common. If you went in any cave in Arkansas, you'd probably find at least one of those bats on the ceiling. The upper right here, you can see that at the caves where we've monitored their populations, and these are generally caves with 100 or more tricolored bats in them, the population was relatively stable, maybe even grew a little bit in later years, although sometimes your population estimates do go up right before a crash when white nose occurs, because some of the bats tend to move to areas of the cave that are more accessible to people when they get white nose for various reasons. But their populations have dropped by almost 90% since 2010. A couple of our other species, the Indiana bat, which has been federally endangered for quite a while, their populations have been kind of stable, but have been slowly declining in Arkansas, even before white nose hit. We're kind of on the very southern edge of their range. And, some of our caves just may be a little bit too warm for them to do well under current conditions. So their populations were falling. There is kind of a bump in populations in recent years, but that's really because we kind of discovered a new population of them in a cave that we hadn't seen before. So that kind of drove up the overall numbers. In most of the caves with Indiana bats, their populations have been declining, even more than previously shown. With little brown bats, which are state endangered, like I mentioned before, we we're also on the edge of their range, and we never had very many of them here, probably never more than a couple thousand. But since white-nose hit, their population has just declined crazily, such that in 2019, I think we only had 11 of them in the state that we were able to find during our cave surveys. So they've definitely been hit hard. And the little brown has kind of been the poster child of white-nose syndrome across the eastern United States, because in areas where there were once incredibly common, you would see large numbers of them, tens of thousands of bats in the caves. And you just saw the impact with that species first. Kind of on the good news side of things, gray bats have actually been increasing fairly well for quite a long period of time. We've implemented various conservation measures for gray bats, Indiana bats, and Ozark big-eared bats for a number of years, and gray bats have responded quite well to those conservation efforts, and have even been increasing in the face of white-nose syndrome. That's pretty representative of what's been seen across their range. And part of what we think's going on with white-nose is that the smaller bats get hit the worst. The tricolored bat is pretty small. Little brown bat's kind of small to medium size for a bat. Gray bats are a little bit bigger. They do well. Same with those big-eared bats. So the bigger bats seem to be more resistant to, or better able to handle having to expend a lot of energy in the winter to get up and deal with white-nose if it gets on them. And gray bats they were being considered for being taken off the endangered species list before white-nose hit, but we wanted to play things a little bit safe at that time and decided to wait to see what happened with white-nose syndrome. But now they're doing well, and the Fish and Wildlife Service is kind of evaluating their status and trying to decide what to do with them. Those big-eared bats have been just kind of relatively stable. They've been in caves with white-nose syndrome almost since the very beginning, and it just does not seem to bother them. Kind of one of the related subspecies, the Virginia big-eared bat, has been increasing in the face of white-nose. So again, the big-eared bat seems to not be bothered by the fungus. It tends to get up and move around a lot in the winter anyway. So if white-nose makes it get up, that doesn't seem to bother it. It's going to move around anyway. So it's dealing with it fairly well. Now, not all of our bats can be monitored in caves in the winter. Some of them, like the northern long-eared bat, will just go in a tiny crack and crevice somewhere where we hardly ever see them in the winter. So for them to figure out what's going on with their populations, we have to look at them in the summertime. And one of the ways we'll do that is try to catch them in what we call mist nets. And here you see some people setting up a net across, I think it's the Buffalo River. And these are just very fine nets that the bats can't tell are there, even using their echolocation abilities, which is what the main thing that lets them fly around at night. They don't see the net and they'll fly right into it and get caught and tangled up. And the U.S. Forest Service has been sponsoring a research project with Arkansas State University for a number of years now, looking at bat populations across the forest to see, if their management activities are having any impact or could have an impact on their populations. And this chart here shows the number of total bats captured on the top line in gray here, the total number of northern long-eared bats captured, and how many nights, or basically the measure of how much effort they put into trying to catch bats here in the middle, in the dark line. And as you'll see over the years, northern long-eared bats were made up a fairly decent number of the ones that were captured. They'd catch 200-300 a year. In general, so they were pretty common. Once white-nose started to hit, the northern long-eared bat populations just started to drop and drop and drop. In the last year, this particular data set, 2018, they only caught 3. While the overall bat captures were still pretty high. They were still catching a lot of bats, but the number of northern long-eared bats dropped off a lot. So we think they have been hit hard here in Arkansas. We're just going to be starting a project to try to assess the impact of white-nose in the Ouachita National Forest starting next year. So we'll have some information on what's going on in the rest of the state at that point. Another species that we can't really see very often in caves is the small-footed bat, which is our tiniest bat here in the state. Normally they'll live in cliff faces in the summer, or just giant piles of boulders and rocks on the side of a ridge. And So in those sort of habitats, it's really hard to come across them. We don't catch them very often with our mist nets, but some folks in Tennessee a while back discovered that they would use these little vertical crevices in the side of concrete guardrails on the sides of bridges. Presumably, these little crevices look a lot like a crack in a cliff face to them. And so they'll use those little crevices as maternity colonies and stopover points. So we've been monitoring them here in Arkansas since, we started in 2013, but we've had really good data since 2014. We'll go there in the summer. If I see any bats in that crack, I'll use a little metal yardstick to kind of coax them out of the crack, and we'll put bands on them so we can follow them and look at their survivorship over the years. And at most of the colonies used by the small-footed bat, their populations have generally been kind of stable. There are a couple, though, where their populations have declined quite a bit. And we have documented some fungus on them in these colonies. So it is having an impact, but it's possible they're not getting hit as hard because they don't tend to form larger groups. And since they're not using the traditional caves that the fungus kind of likes to live in, maybe they're just not getting exposed to the fungus as often here as in some other parts of the country. So that's kind of a quick overview of what's going on with the bats here in Arkansas. I'd be happy to take any questions.
All right. Does anybody have any questions for Blake? Blake, I'll throw one out there. You mentioned a couple of things, is like susceptibility to white nose, body size, the way they congregate, where they congregate. Are there, understanding this is all relatively new, what are some other factors that might be cause a disproportionate effect on different species, I guess, with white nose syndrome?
Well, there's some thought that some bats have kind of different substances on their wings that might actually kind of prevent the fungus from taking hold and getting established on them. They're still investigating that to see if that's a thing. That might be what's helping the Virginia big-eared bats back east, and presumably could be present on our Ozark big-eared bats here, but that hasn't really been proved yet. But yeah, it's generally the size seems to be the biggest factor. The big brown bat, which is very common here in Arkansas, will get the fungus on it and can see some impact, but it's not being hit nearly as hard as the other smaller species are.
Thank you. Any other questions? All right. Hearing none, seeing none in the comments. I think it was just a good job comment from Matt Warner. Anybody else going once, going twice? Tate and Blake, thank you very much. And to all of our colleagues who tuned in, thank you for being here. Great presentations. If anybody wants to share this, it will be available here in the next couple of days on the Arkansas Game and Fish Commission YouTube page. Thanks, everybody, and we'll see you next month for the September version of the Wild Science webinar. Take care.